Hello boys, welcome back to another session of online biology video tutorials, and this session is meant for class 12th biology students, and this is for 30th July 2021. As all of us know, we are into the debate and deliberation of chapter number 5, Principles of Inheritance and Variation. In this, we have already covered in our previous sessions what is inheritance and what is variation, what is Mendelism, the laws of Mendel's, what we call Mendel's laws of inheritance. We also covered gene interaction with one gene inheritance. We call it as mono-hybrid cross. Now in this session we are going to deal with inheritance of two genes. We call this as dihybrid cross. Di means two, hybrid formation takes place, and the crossing of the two, hence dihybrid cross. So it is a cross between two parents differing in two pairs of contrasting characters. That's why it is called dihybrid. Example, cross between pea plant with round shape, as you are seeing, pea plant with round shape and yellow color. So two characteristics are there, round and yellow. So since it is dominant, how do you say that round yellow is dominant over wrinkled green? When we count the number of seeds that are formed, like actually Gregor John Mendel, with his lot of patience, he could count. So based on the numbers, he understood that round yellow seeds were more in number than wrinkled green. So whichever numbers are more, that character would automatically become dominant character. And the one that has got low numbers, less numbers, that character would become recessive character. So there are two contrasting characters. One is the shape of the seed. Round seed is dominant over wrinkled seed. Okay. Again, why? Number of seeds that are in round shape are more than the wrinkled ones. This is very common experience. Even in your home you must have seen. Whenever you are purchasing green peas, you must have seen the round ones are predominantly dominant over the wrinkled ones. Similarly, yellow is dominant over green. So round yellow, both the characters are dominant over the contrasting characters, namely wrinkled and green. So when we give the genotype, as I said in my previous session, for round the spelling starts with R-O-U-N-D. So the first letter of that is R. Since it is dominant, we pick up the capital letter. Since it is homozygously dominant, we say capital R, capital R. Two alleles are responsible for that particular character. Similarly, color yellow. Since it is dominant and it spells with Y-E-L-L-O-W, Y should be capital because it is dominant. Since two alleles are involved, we are writing it as capital Y, capital Y. So overall, phenotypic character round seed with yellow color is represented genotypically as capital R, capital R, capital Y, capital Y. So let me write here, for round it is capital R, capital R. For yellow it is capital Y, capital Y. Versus, okay, versus, it's not multiplication, it is cross. Since I said, if you may recollect from your memory that whatever alphabets we pick up for the dominant character, corresponding small letters only we should pick up for the recessive character. Like here, round is capital R, capital R. So you ought to take for wrinkled small r, small r. Don't take small w, small w. You will be confused. So similarly, for yellow, we are taking capital Y, capital Y. because it spells with yellow. I said YELLOW. Corresponding letters, alphabets is small y, though the phenotypic character is green. Don't write small g. So what you have to do, whenever there is a problem related to genetics, you must follow certain precedences that are set in by the geneticists. The precedence is that for dominant we pick up the alphabet capital letters, and for the recessive one corresponding letters only you must pick up. Like you are picking up capital R, the recessive character should be represented genotypically with small r. So overall, if you see in this, since two characters are involved, we say dihybrid. So round one is parental generation genotype would be capital R, capital Y versus small r, small y. This is P generation. P generation means parental generation. So this is the phenotypic character as well as genotype. So we have represented both phenotype as well as genotype in this fashion. I hope I made it very clear to you. Going ahead, when we go for the cross, the parental generation, we know the phenotype, homozygous round yellow, homozygous wrinkled green. And genotype also we know capital R, capital Y versus small r, small y. When we go for the crossing of these two, what type of gametes will be likely to be formed? From this capital R, one capital R, capital Y, one capital Y. Why you are saying only one capital R, one capital Y? You know, this is meiosis. You know, the background for this you must understand is meiosis. So meiosis is meiotic 1, meiotic 2. So in that, when this division takes place, virtually you have two gametes having genotype capital R, capital Y. Similarly, in this case, small r, small y. That means I can say in this way, capital R, capital Y, small r, small y, small y. Now selfing takes place, and this we call it as F1 generation, first filial generation. And when selfing takes place between the F1 generation, the gametes that are formed would be capital R, that is capital R, capital Y, small y. This capital R and this small r. So capital R, small r. This capital Y and this small y, this. So in the F1 there is selfing. When selfing takes place, what are the possible gametes that are formed in F2 generation or say in meiotic 2. So from this plant having the genotype capital R, capital Y, small y, the possible gametes that are formed would be 4. The 4 gametes will be having capital R, capital Y, that is this combination capital R, capital Y. Second one is capital R, small y, second combination. And third one is small r, capital Y, like this. And left out one is small r, small y. Since the genotype of this parent also same, the gametes also would be same like that of these 4. Hence, we have 8 gametes, 4 from each. But the genotype is almost same. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Now let's see the crossing of these two. So you require the Punnett square for this so that it will be very easy. So 4 into 4, the mathematical calculation is so simple. 4 gametes on your x-axis, 4 gametes on y-axis, 4 into 4, 16. So you have 16 offsprings that are formed. And let's see what will be the genotype, what will be the phenotype. Now let's see the crossing. If this gamete of one parent combines with this gamete, what would be the resultant offspring? Capital R, here you are having capital Y. So capital R, capital Y. What would be the phenotype of this? Since it is fully dominant, capillar, homozygously dominant, round, homozygously yellow. So fully dominant. Now this zygote combines with this. So capital R, round, phenotypic, capital Y, small y. Even though small y is there, law of dominance, remember law of dominance, dominant gene capital Y is there, so the fellow becomes yellow. So round yellow. Similarly, when this gamete combines with this, what would be the combination? Capital R, small r. From this gamete, capital R. From this gamete, small r. So capital R, small r, and capital Y, capital Y. So again law of dominance, capital R, round, yellow. Now what is left out? Last one, capital R, capital Y versus small r, small y. So you get a genotypic combination of capital R, capital Y, small y. Still it is round yellow. Why? Law of dominance, capital R, capital Y. Hence this offspring also becomes round yellow. So if you see horizontally, all the offsprings will be round and yellow. So round yellow, which is the characteristic feature of one of the parents, particularly dominant parent, correct? And next one, we take up the capital R, small y. When this capital R, small y combines with this, you get the first product, capital R, capital Y, small y. Still this is round yellow because law of dominance. Next, you see the new one, round green. See, you have got wrinkled green. This is the new character, new feature, new offspring, new product. That's what we say, law of independent assortment. What we said, Mendel's laws 3. Law of dominance you understood. Because of the presence of dominant gene, dominant character will be formed. Law of segregation you have seen. Just now you have seen the capital R and small r breaks off. Capital Y, small y breaks off. Capital Y breaks off. So the alleles segregate. And after segregation, what happens? They have freedom to combine with anybody. So thanks to this feature, when the alleles combine in different form with a lot of freedom, varieties are formed. See, we have taken round yellow parent crossed with wrinkled green, right? Remember? And these are the new ones, round green and wrinkled yellow. So Mendel at that time was not knowing how is this possible, from where these characteristics are coming, from where these new combinations are taking place. He only said factors are responsible. Something called factors will be present inside the body of an organism, and those factors are responsible. But now with the latest development of technology, particularly genome structure, we understand that it is because of the genes, and the genes combine in a new fashion. Look at this, capital R, small y is a new combination. Since this is a new combination, lot of freedom must be there to combine. That is why Mendel called it law of independent assortment. And not only they independently, freely combine, they assert the phenotypic character. So round green is a new one. And next one, capital R, capital Y, small y, round yellow, again parental character. Whereas look at this, capital R, small y. This is law of dominance. Again round green, which is a new feature. Third one, capital R, capital Y, round and yellow, parental character. Next one also parental character. Next one, small r, capital Y, small y, wrinkled yellow. See, round yellow is the feature of the parent, but first time we are getting a new character, new combination. Wrinkled yellow has come. Similarly, wrinkled yellow 2, wrinkled yellow 3. Of course, this is a parental character, round yellow, and this is the new one, wrinkled yellow, new one, and wrinkled green, the parental one, ancestral. Particularly Ajji's character, grandmother's character, wrinkled green. That's why we say that some of the features in grandchildren will be the copycats of the grandparents. That is the reason why we say even physical appearance. Sometimes, your neighbors, your relatives will be speaking that you look like your Ajji or Ajja, your grandfather or grandmother. From where this character has come, this is the combination, right? You understood? Now overall, if you see, not just two round yellow is the parental character, wrinkled green is the parental character. But what are the new characters that offsprings, children have got? Round green is a new character, wrinkled yellow is a new character. So this is the magic of dihybrid cross. Okay. Now look at this. On observing F2, Mendel found that the yellow-green color separated in a 3:1 ratio. Round wrinkled seed shape also segregated in 3:1 ratio. Overall, if you see, how many round and yellow? 9 are there. You can count. Okay, 9 round yellow. How many round green? 3 will be there. How many wrinkled yellow? 3 will be there. How many wrinkled green? Just now we said only 1. So the dihybrid F2 phenotypic ratio universally fixed to 9:3:3:1. Okay. So this remains fixed 9:3:3:1. This ratio can be derived as a combination series of 3 yellow is to 1 green with 3 round, 1 wrinkled. So 3:1 into 3:1, 9:3:3:1. This is the universal structure. On observing F2, Mendel found that yellow-green color segregated in a 3:1 ratio, round wrinkled seed shape also segregated in 3:1 ratio. Okay, as far as phenotype is concerned, right, fine, 9:3:3:1. What about genotypic ratio? If you see the genotypic ratio, it is like this. Capital R, capital Y, 1, capital Y, this 2. So another capital R, capital Y, 2. Capital R, small y, 4. Capital R, small y, 1. Capital R, small y, 2. Small r, capital Y, 1. Small r, capital Y, 2. And all recessive 1. So 1:2:1:4:2:1:1. This is genotypic ratio. Okay. Overall, you can see totally it should be 4:4. I said 16. It cannot be more than that. So it is based on the results of dihybrid crosses. And based on this, law of independent assortment has come. Segregation and reassignment. It states that when more than one pair of characters are involved in a cross, factor pairs independently segregate from the other pair of characters. So that's how the character segregated and new combinations have been formed. These are the various segregation factors. And the concept of dominance, every gene contains information to express a particular trait. In heterozygotes, there are 2 types of alleles. Unmodified or normal functioning allele, it is generally dominant and represents original phenotype. Modified allele, it is generally recessive. So you got here, you can see allele of the brown eye color is unmodified allele, whereas allele of the blue eye color is modified allele. So there is modification also. And the concept of dominance continues. In the law of dominance, consider a gene that contains information for producing an enzyme. Normal allele of that gene produces a normal enzyme. Modified allele is responsible for the production of